Hello everyone, welcome back to Science on Trial and Error. I'm Kasia Kowalska and I'm excited to introduce you to yet another amazing scientist. But before we get to that, I'd like to apologize for the delay with the premiere of this episode. Some unforeseen health issues have kept me preoccupied, and I needed to take care of this first. Thank you for being patient, and let me reassure you, this episode was worth the wait. Today my guest is Laura Burnett. She comes from England, and that is where she obtained her master's degree in natural sciences at the UCL. Laura currently pursues a PhD at IST Austria in the group of Max Joesch, and she's studying how visual information is processed in the brain to control instinctive behaviors. But besides being a neuroscientist, Laura is also an artist. She's creating incredible pieces of art using different media, and she contributes her creative side to science communication projects. Make sure to check her Instagram to get a sneak peek of her works. I absolutely adore this quirky girl, and she has always inspired me with her strength, positive attitude, and outgoing personality. I'm very grateful that we had a chance to talk honestly about the ups and downs of her scientific life. We also had a fair share of laughs during the recording. She's definitely the biggest David Attenborough fan you'll ever meet. Please welcome Laura Burnett. Hello, Laura. Thank you for accepting the invitation to be the guest on my podcast.
No, it's great.
How are you doing?
I'm good, thank you. How are you?
I'm fine. I'm very excited. I was thinking to have a sort of a starting point in the discussion, which should be quite easy. Mm-hmm. And I wanted to ask you, what is it that you listen to at work, whether it's music or something else?
Oh God, that's a good question. I mean, it depends what I'm doing. Like, if I'm doing a very monotonous task in the lab or something that I'm quite used to by now, like surgeries or something, then podcasts or the Hamilton soundtrack, which I went through a phase of just listening to that on repeat, which is quite embarrassing, but it's wonderful. But normally podcasts.
Okay. So how about we talk first about your current work. Mm-hmm. You're a PhD student in the group of Max Joesch, who works on neuroetiology. Is that the word?
Yeah, that's it.
So can you explain maybe a bit what are you working on and what is the group working on?
So Max named the group neuroethology, which is basically just the neuroscience of behavior, which is how the brain is able to control and mediate different behaviors. And in our group, people work with both flies and mice. So we look at two different animal species, and we're specifically interested in how vision, like visual information, is used to drive behaviors. Mm-hmm. Yeah, we use a lot of different tools, and like the behavior that I'm actually interested in is a visually evoked defensive behavior.
Okay.
So it's basically, I'm interested in the circuitry in the brain that is involved in how an animal, for instance, a small prey animal like a mouse, if it sees a dark shadow overhead, which could be like an overhead predator, an owl or a bird that's about to approach and attack it, how they process that visual stimulus of the shadow as a predator, and then how they mediate some sort of defensive behavior, be it freezing or fleeing to an escape, like doing an escape to a shelter or something like that.
So is this an innate behavior?
Mm-hmm, yes, exactly. So it doesn't need to be learned. And it's interesting because humans, like human babies especially, like if you show them a looming, basically a dark expanding dot, which looks as if it's coming towards you, like they show this sort of reflexive.
Oh, really? So
yeah, like
it could be evolutionary conserved.
No, definitely, yeah.
Oh, that's really
and flies also show the same like jumping behavior away from a looming stimulus.
So you have an environmental, like experimental setup to track this. Mm-hmm. So you definitely track the motion of the animal.
Yep.
Do you have a way to also track their brains and like record what is happening in their brains?
So at the moment I do the recording in the brain in what we call like a head fixed situation. So the mouse, it can run, but it's sort of fixed in one position. It's running on a ball, and we have this like screen around, completely surrounding it, where we can present the stimuli. So it's sort of like a planetarium-esque situation for mice.
Okay.
It's an immersive,
It's
semi-virtual
virtual reality
virtual
kind of
reality
situation. Okay.
Not quite that fancy, but still it's cool. And with that, I use what's called a silicon probe.
Okay.
So it's a very, very small probe which has 32 channels which record neural activity like extra outside of the cells. I'm not like recording patching or anything like that. So we can basically listen into what lots of cells in the brain are doing at the same time. And I'm looking at one particular brain area called the superior colliculus, which is cool. It's a highly evolutionary conserved region. It's found in all vertebrate species. Humans have it as well, and it's a really cool brain area because it receives input directly from the eye, so it has direct sensory input. People debate about it, but it's like could potentially be the most important visual brain area for the mouse. In humans, it seems it's less important because another visual pathway has sort of taken over. But it's predominantly involved in these sort of innate and intrinsic,
like the
visually,
fast reactions.
Exactly, very fast reactions. Because it has this direct input from the retina, then it also receives information from other senses like touch and auditory information.
Oh, that's interesting.
And all of these senses are like retinotopically mapped. So this means that like the position at which the inputs from the eye terminate in this brain region are arranged in the same positioning as the visual field. It's sort of layered like an onion. So the visual input comes into the top layer, and then you have these sort of inputs from different sensory modalities coming in this same ordered way, like aligned with the visual scene, but in deeper layers. And in the very deepest layers, you have motor output layers. And then it has these motor cells that then send information to other regions of the brain and the spinal cord, which say, okay, you need to move your eye or your entire body to this location. One of the first things that people realize that this brain area is involved in is like orienting towards objects or away from objects. But
also your experimental setup sounds really cool. So I guess it requires also a bit of like engineering kind of aspect to your work.
Yeah.
Was it really hard to get into this part of the work?
It was definitely new. I definitely hadn't done anything like this before. And also because the questions that I was asking and my ideas changed so much over time that I think I've built like 5 different experimental setups, like all along the same lines, but I needed it to be slightly bigger in this case, and I needed this to be slightly different. And for instance, I have a projector that projects the visual stimuli onto the arena. First I had 3 projectors because I had a domed roof and I wanted it to be like like an actual planetarium.
Yes.
And then I realized that wasn't working, and then I had to change to a box. And then, my God, that is really trial and error. That was, but it was really, really fun. And I'm very happy with the setup I have now. And now I've made it so that it's very modular in a way, which I think is very useful. If you're ever, planning on building an experimental behavioral setup, make it as modular and as adjustable as possible, because
you never know.
Yeah, you never know, and it's been very fun learning about that.
So I think what I also read on your website is that you're using optogenetics in your experiments, right?
Yes. So I'm also so it has been shown that if you activate the cells in this deepest layer of the superior colliculus, if you activate these deep motor cells, the animal, so they did this in small, like a mouse or something, will move either its head or its entire body or its eyes to that location in space, depending on how strongly you activate it. I've been trying to do that in my mice because in a way if you use a strong enough activation of the cells, it's sort of
reproducing?
Yeah, reproducing the effect of the looming. Then the animal will think that there is an object there, and then they should behave accordingly, which is what we see actually, which is quite cool.
So you actually see that the cells really reproduce the position in space. But it's interesting because, Yosman was just here talking about the grid cells and, how the brains can encode the spatial position. And it's like a cell that corresponds to a position in space. And now you're seeing this in a different region, but it's kind of comparable situation in a way. Like that the exact cell corresponds to a area in the retina which would correspond to position, right?
Exactly. I think that's the thing. It's not because I think in the hippocampus, and what she's talking about is more egocentric position in the environment, whereas this is more like objects and their position within the visual scene,
within the visual picture, like the retina,
exactly,
seeing it,
like retina-centric.
This is really
but it's really cool, isn't it? Yeah.
Yes.
And it's really interesting from a developmental perspective as well, which is not what we study in the lab, but lots of people have studied this before, like how the visual input in the superior colliculus then helps with the development of the maps underneath in the different senses. So like they've found that like if you disturb the inputs of the retinal cells to the top layers of the superior colliculus, then the layers below receive input from other modalities. They are also sort of messed up and misaligned.
So would people that have retina-derived blindness have problems with this region? Do you know?
So that's sort of what people think. I mean, it's hard to tell because in humans, like I said, people don't think it's as important.
It's a different important level.
Yeah, because so the way that I try and explain it to people is that the common way that people think about how vision is processed in the brain goes from the retina to a region in the thalamus, which is in the middle of the brain, called the lateral geniculate nucleus, and then that sends the information onto the visual cortex. Yeah. And in humans, this is the predominant visual pathway. And then in the cortex, there's a lot of processing of visual information going on. And this is more like conscious visual information. Whereas the superior colliculus, it seems, is more to do with subconscious visual information.
Like really fast reactions. They're the ones that are...
If someone throws a ball at your face and then you sort of try and dodge to avoid it, this is more like your superior colliculus is like being like, whoa, like alert. Ball coming at your face, or like there's a car gonna hit you or something, the mouse is being like, oh, there's a predator coming to attack me.
Interesting that still the humans kind of maintain it, no longer like there's so many predators against humans. I guess you could expect it to kind of decay a bit, like this sort of information processing, but
people think it's also to do with like emotional processing. Also importantly to add is that the superior colliculus develops before the cortex, even in humans. So there's a lot of research that's been done showing that the superior colliculus is also needed to develop visual processing in the cortex
already during early development.
Exactly.
I see.
Which is really cool. But there's also a condition called blind sight in humans. Blind sight is basically when people that are blind, basically they can't consciously see anything, and it's normally due to a problem with their visual cortex.
Yes.
And these individuals, if you put them in a corridor with obstacles, they will say that they can't see anything, but they can avoid the obstacles.
Oh, but then if there was a different region that processes this kind of
Exactly, it's just like unconscious visual processing. And people think that this could be because the superior colliculus is still functioning and that they can subconsciously still see things. I love it. It's a really cool area. I think
it's great.
It's been a little bit neglected, I would say, for a few years. I think people have become very obsessed with the cortex, and now it's making a bit of a comeback. So
no, it's good. It's good, and your work sounds really exciting, and like things are working well. And
I mean, ups and downs.
Now, I like to talk to my guests about how and when or where their love for science started. And I think everybody has a very different story. Yeah. Because your parents, I don't think they are really like science involved, right? And
no.
Is anybody in your home actually a scientist, or are you the only black sheep in a family who went into the direction of the lab?
I love this idea of black sheep. I didn't know a single scientist. Like my parents are definitely not into science at all. I didn't know any scientists. I mean, I grew up in a small village, so people had normal jobs, and I think it wasn't until I got to university that I realised that, you can do science for a living, like that was an actual job. Because when I was a kid, like even at school, I thought, if you're going to do science, you're going to be a doctor. Like that was, a doctor or. I didn't even think I knew like you could do like. I mean, I was like, maybe you can be like an astrophysicist or something like that. But it was. I definitely was very naive about things. But I always loved animals and I always loved nature. So that was one thing. Like, I mean, I lived in the countryside, so I spent a lot of time just in the garden and in woods and like picking up bugs and planting like apple pips, and then I grew trees and then they got infected with a mold, and then I just loved all of that.
So the curiosity was definitely there.
It was definitely there. It was very wild, I would say, and unrefined.
Was it also in part driven by Sir David Attenborough's
sure,
adventures and programs?
Definitely. I think that was...
So did this love start really
with
early
David?
on?
I think yes.
Okay.
Yeah, anyone, as you know...
I think anybody who knows you...
I'm a huge fan of David Attenborough documentaries. I mean, the BBC Nature, department in general. Because I was actually born in Bristol, where the BBC Nature, HQ, at least they were there. I don't know whether they still are there, but I hope so. They're still there. And as a child, I was like, one day I'm going to work for BBC Nature. I think that was my first dream. I think I was like, I'm going to be David Attenborough. Not quite sure how that works, but you know what I mean? I was like, but again, I had no idea how someone ever gets to be, how that happens and what sort of... I think I didn't really think about the future that much as a child. I was just sort of doing what I like. I remember having to do a project when I was in year 8, I think, so maybe 12 or 13. And we had to choose a natural history project. And I lived next door to it. Yeah, I know, fun. I lived next door to this small wooded area. So for I think it was 2 weeks, my dad got up really early before work, and me and him just went and sat in this wood, sitting there. It was such a me and my dad situation, sat on 2 camping chairs, watching, and we saw a deer, we saw some really cool birds. I just wrote down what we saw and noted.
That's really committed, and that seems like a good start to be a proper... reporter for BBC Nature. You get this patience to practicing the patience to watch and observe animals in their natural habitat. I honestly think that most people in the UK that are into biology have been inspired at least in part by the BBC Nature documentaries, because they were incredible. They still are incredible. I agree. I mean, I think it's a worldwide phenomenon, but of course we were we knew these documentaries in Poland, but we didn't really know David Attenborough so well, because in Poland there was another person that was reading the whole background. So we all knew those. They were super exciting, but only after actually I met you, I realized okay, there's this special person that is actually behind all of them, and it was really cool to learn. So your parents, even though they weren't into science, they weren't doing science, they were actually very supportive of you doing your own thing.
I think, my parents are incredibly supportive. They think I think I'm a bit crazy probably, but they love me. My mum once got me this badge that says, "I'm not weird, I'm gifted," which I think sums up what my family think about me. But no, I think, they just let me do whatever I wanted to do. And my dad was always in the garden. I mean, he also grew up in a very rural area, so we had a vegetable patch, and we used to... I like to think that I used to help him. Probably if you asked him, I was more of a hindrance, but...
You were from a small, town? Or
village, really not a town.
Okay, and then you went to school nearby. Were your classes at school focused on going into science direction, or were they more general education? Did you have to choose the direction in which you want to go at some point, or?
I mean, so my primary school was just a very small local village school. We had 13 people in my year, so it was, really... Tiny. So it wasn't really... It was very general. I think they just... I mean, it was a great school, and I loved my primary school. And especially... So my headmistress actually taught me maths, and I think she, like, now when I look back, I'm like, she was such an inspiration, and sort of she really pushed me forward even from such a young age. Like,
oh, that's great.
I always look back and think, wow, I could have gone so differently.
Yes.
Yeah, and then I moved to like a bigger school for secondary school, and it was an all girls school, but it was very academic, so we had to like take an exam to get in and stuff. Whatever you wanted to do, you could focus on whatever you desired.
I see.
Yeah, which was cool.
So were you still into maths at that point, or?
I mean, so for AS levels, I took further maths, biology, chemistry, and Spanish.
I see.
So I tried to keep it broad because I had no idea what I wanted to do.
So then you moved to London for your studies,
yes.
right? You were studying at UCL.
Yep.
And what was the actual
course?
Yes, what was the main?
So I did
course?
natural sciences because, as you'll discover, like a theme of my life is that I didn't want to narrow it down, so I just kept.
Amazing.
It was actually my friend's mum who was like, "Oh, Laura, there's a course called Natural Sciences where you can just keep doing all of the sciences," and I was like, "This is ideal."
So was it a joint bachelor's and master's program?
Yeah, this was a joint MSci thing, so it was 3 years of bachelor's and 1 year of master's.
Yes.
And basically in the first year you start doing all of the sciences, and then you have to drop a few for the second year, and then in the third year you specialize, and in the masters you just do I specialized in neuroscience for my masters.
So it wasn't really always neuroscience in your head as a future path?
No, definitely not. Neuroscience happened sort of completely by chance.
Okay. So was it because you also went to Melbourne,
yes,
right?
I was.
It was like a research internship.
I know, I did my entire third year in Melbourne. I applied to the study abroad scheme. Mm-hmm. Because I don't know, I just always had, now I'm living abroad as well, so I obviously had something in me that was always really interested in living abroad. And I think because I did Spanish, I continued doing the Spanish when I went to university as well. I'm going to go somewhere where I can speak Spanish, it's going to be great. I'm going to improve that and do some science, and it'll be great. And then obviously my university were like, the only places you can go are places that speak English. And I was like, excellent, cool. And then I just happened to, I looked at what courses were available, and Melbourne seemed to have a really good, offer a lot of the courses that I was interested in. Off I went for my third year, had a wonderful year. And that was actually, how I first got into neuroscience, really.
Oh, really? So the project really changed your, started your love for neuroscience.
So basically what happened was, I love Australia, and I think it was amazing, but they were definitely a lot more relaxed with attending lectures, or at least with this one course that I went to, people were very relaxed. Developmental neurobiology, I think, was the course. And I think I just took it because I needed an extra credit, and I was like, oh yeah, it looks sort of nice, I'll try that one. And then this one lecture, I turned up and it was just me in the lecture. It was a small course, there were only 30 of us. And it was this really nice woman at the front who was really funny, and she was like, looks like it's just you and me today. And I was like, okay. She was doing interneuron migration in the cortex. And I was like, wow, this is really cool. And she was telling me about how interneurons migrate in the brain, and how the place in the brain that they come from looks like the Batman symbol. And I was like, this is amazing. And we got chatting, and she was like, oh, well, you should come see my lab if you want. And I was like, that would be great. So I went. But that was my first ever experience in the laboratory sort of thing. And then I got in contact with other labs at the University of Melbourne to be like, oh, would you take me for a small bachelor's research project? And this woman called Professor Heather Young wrote back to me, and she was amazing. I loved my time in the lab, and they were working on the gut nervous system, the enteric nervous system. Again, I had no idea how the job worked, what people do in their day-to-day time, and it was really cool seeing so people they meet, they discuss ideas, they come up with suggestions for experiments, they do them, they present them, it was.
I love how random it seems.
Oh, everything is so random.
There's so much lack on your path to put these right people there so that you can be actually, because you have really a huge passion for science, but the thing is it was like so not focused at the beginning, right?
Yeah.
You were just like interested in everything, but like you didn't even know what is out there. So then you were sold for like nervous system.
Yeah, and I was really obsessed with the gut nervous system. So then I went back and did my master's and got in contact with a group at UCL that were working on the gut nervous system. And I still love it, to be fair. But then I got sidetracked and ended up in the brain. I think I'm very lucky because I've enjoyed everything that I've done, and I'm just sort of continuing with my rolling through great scientific adventures.
But I think this comes also from your like openness and like, because you know some people they find something that they really like and they just like very persistently go in this direction, and with your case it seems like you were just always so curious about so many things that it just makes it also easier for you to go into the new things. Like you are like very open to the new opportunities, and it's super. I think it's very important in a scientist. Like of course you can be very focused, but I think it's good to have your mind open to like a new region or a new method, or it's just it can bring you a lot of joy and a lot of surprises.
No, definitely. I mean, I'm constantly reading. Like, I love reading popular science books outside of my field, just because it's sort of like when I recently read this book, Entangled Life by Merlin Sheldrake, on fungi, and then I was like, oh my god, what am I doing? I need to work on fungi. Like, that's just the coolest thing ever right now. And then I was like, no, Laura, you're doing a PhD in neuroscience.
At some point, you can still go in this direction. So you did your master's working still on gut enteric system,
exactly.
At UCL,
At the Institute of Child Health.
Were you still interested in like development, or was it more disease related research?
I mean, it was both. So I mean, the group was really cool, like in the way that it was set up, because the institute is linked with the Children's Hospital, and so our group was actually co-run by a clinician and a researcher.
Yeah,
like we were getting direct information from the doctor that was doing the surgeries on the children and stuff, and we were specifically, like, the group were looking at this disease called Hirschsprung's disease, which is basically where the nervous system in the gut doesn't fully form
during the pregnancy.
And it's lethal in babies. Like, they have to treat it very quickly. Like, if you don't treat it, then it is lethal. So the group were looking into, like, okay, well, how is the nervous system developing? And we were looking at, you know, how is it colonizing the gut, because the gut is already formed, and then the nervous system cells have to, like, sort of migrate in and colonize. And lots of they were doing lots of really interesting things with, like, stem cell therapies and things like that. So
you were, like, really into this topic, and then you were you looking for PhDs, or were you not so sure whether you want to do a PhD? How did it work?
So I think it seems a long time ago now. I'm sort of, but so I was doing the masters, and I really loved my group, but I think they were, like, I sort of got told that, you know, funding was not secure and stuff, and that maybe they wouldn't be able to offer me a position and stuff. So I was like, okay, I will, you know, do an internship. And my idea was that maybe I still wasn't 100% sure, being me, what I wanted to do. And so I was like, I will dabble around, try and see if I can get different, like, maybe a technician placement somewhere, and see if I can try out things in different labs and see what I was interested in. And then I found this internship scheme for IST Austria, where we now are, and I applied. I was just like, why not? Like, let's give it a go. And I was really interested because it was like a new place, and there was lots of interesting science going on, and there were, you know, and and then I got accepted. So 2 weeks after I finished my last exam at UCL, I went to Austria for the first time in my life, and was like, sure, speaking no German. Always just, you know, why not? And then it was really fun. I had a great time. And then they asked if I wanted to stay for the PhD, and here I am.
So you were doing a project in the group of Simon Hippenmeyer, who is working on this very novel technique.
Yes.
Called MADAM.
Mosaic analysis with double markers.
Yes.
In the cortex.
So how did you like it? How did you like this experience?
I mean, I think I was, again, exceedingly lucky. because so I arrived and I was working with a really great postdoc called Rob, and Rob was basically at the end of this project that he was doing, and he was wrapping up, and he basically just needed some help, and I was there, very willing and very keen, and was like, yeah, this sounds great, and we got on really well together, and he was a really good supervisor and stuff, and it was great. It just seemed very dynamic, like things were going, we were doing stuff, I could learn all about this new genetic technique.
Yeah.
So it was nice that there was a lot of trust, and I learned a lot during that internship.
So it was a summer internship, right?
Yes.
And then right after you jumped into a PhD.
Yes, it was 3 months, I believe. It was
okay.
Yeah, 3 months. And then I finished in
That's crazy.
September. I started. I think I went home for a week to graduate, and then I came back, and then, yeah, I know. I also think, yeah.
That's incredible.
I have like my dad's voice in my head just being like, don't lose momentum, which, you know, reflecting now, I'm like, you know, sometimes it's okay to lose momentum. But
I think so. But I think, I mean, again,
yeah,
it was not planned really, but somehow turned out to be really good, I guess, right?
yeah.
Okay, your studies at UCL had this period of being very interdisciplinary at the beginning, and
Yeah.
here again you had a lot of interdisciplinarity in the first year.
Yes.
And also there were rotations.
Yeah.
Did you actually like the fact that there were rotations? Was it fun for you?
Yeah, it was really fun. And I mean, I think that was a big part in why I decided to join, because to be honest, I felt very young when I first arrived. Like, because a lot of places in Europe, you know, they have 4-year undergrads and then 2-year masters, and people.
So how old were you when you were starting the PhD?
I was 22. Yeah, I think so.
Yeah,
22.
That's quite young. Yeah,
Yeah.
that's true.
Yeah, and I mean, I had done things in the lab, but you know, I think for a PhD, like I was like,
It's
okay,
a
you need to.
long-term commitment.
It's a long-term commitment, but also like, you know, you need to come up with your project. You need to. And I was just like, my God, I'm not experienced enough for this. You know, imposter syndrome hitting hard, and I was like. And so at least I was very like, okay, well, I have the rotation system. If I don't find anything, or if it's not what I want, I can leave and look for something else. And then I found something that I like. So
That's great. No, I think it's good. Did you stay though within the neuroscience? I think so. Yes.
I went to the 6th group.
So so you did
cell
something
migration and
different.
stuff. But that's sort of similar to how the gut nervous system develops. Yeah, exactly.
But that's good. You've explored a bit outside.
No, it was really nice.
And
also, I'd never worked
yeah.
with cell culture before.
But also you never worked with behavior, and now suddenly
That's
here
true.
you were doing, like, starting a project in behavior.
Yeah.
So how did it work? You decided to join because your rotation project was interesting, but then, as you said, you had to develop your own idea.
But I think that, the idea was, to be honest, was difficult. I mean, the main selling point for me, and what really, like, I was like, wow, this is really cool, was I sort of got won over by systems neuroscience. And I loved the techniques, and I don't know, there's something about seeing neural activity, like activity in the brain, and I was like, wow, this is cool. And I just felt I had so much to learn, like from the rotation that I did with Max, I learned so much. We were building, it's a very loose definition. Max was building, and I was sort of supervising and handing him screwdrivers and stuff. The 2-photon microscope for the lab.
Yeah.
It was, again, something that I just loved it, because I felt like it was an incredibly multidisciplinary part of neuroscience. You know, you're sort of engineering things and building things. There's still the genetics, like there's the animal behavior, there's the viruses that you need to use. There's so much involved.
So the aspect of just still being able to learn so much was also important to you, to kind of just develop more and like grow in your project as well.
Definitely. Because I think, and I still think it now, like, I'm really glad that I've learned all these techniques, because I think you can apply them to many different aspects of science. I mean, definitely different aspects of neuroscience. And, you know, if I want to go back to gut nervous system, it's always cool to have people that know how to record in different things, you know.
Yes, definitely.
And I think I feel a lot more confident now trying new things.
So exactly, with this kind of project, a lot of things may not work.
a lot of things didn't work.
So was it hard for you to deal with the things not working, or were you actually like you were expecting it, so you were treating it lightly?
I felt like an outsider when I started because I obviously hadn't worked in it before.
Yes.
So I was expecting to fail. Yet again, I think most people that sign up to do a PhD are sort of born perfectionists and have got to that position sort of swimming their way through school and university and getting things right all the time, and you're like, yay, everything's great. I just need to read this and learn this and then everything will be fine. And then you get to PhD and like you can read and you can learn as much as you want. Like
it's always going to happen.
Failure, so much failure. And I think at the beginning, it was hard. It was really hard. I think at the beginning you sort of you buff it off with your enthusiasm and you're like, it's fine, I'll just try the next again. But after like months and months, and when you're not sure why you're doing something, and then it's like, because I changed my question like 2 years in. I was trying something for ages, and then I just came to this point where I was like, this isn't going to work. Like, mainly because at the beginning I didn't know enough about the technique and its limitations. And then,
to make an informed decision.
Exactly. I thought it could do more than it could.
Yes, I see.
And then I was like, okay, I'm not going to be able to do this. Like, maybe, I don't know, if I had 10 years, an infinite amount of time, and, I could do something, but with the tools that were currently available and my time limitation, I was like, it's not worth it. So then I decided to follow a new avenue, and now it's a lot more fruitful, shall I say.
I mean, sure, it sounds like the project is going really well, but it takes a lot of courage and also a lot of determination to change the project. So how do you think you managed to keep your motivation up? How did you deal when you were at this point?
Oof. No, I would just say that I didn't deal very well, I think, is what I was... I was very sad. Yeah, it was very, very tough. And I think as well, moving abroad is a great thing, but it's a very lonely thing as well,
Sure.
And it's... I don't know, I think I'm a very independent person as well, which I think, again, a lot of PhD people are, but, I think I'm sort of stubborn and proud a lot of the times, and you need to let that go if you're doing a PhD, I think, because you need to...
you need to accept certain
I think, so I wasn't...
weaknesses.
Exactly, accept the weaknesses. But I definitely wasn't having a great time. Everything was failing, and I just felt like a failure. Everything is just failure, left, right, and center, and you're just like, ugh. And honestly, the reason that I changed the entire project basically was fluke, a complete fluke again. I'd read a paper.
No way.
Yeah, no, actually, it is the story of my life. But I read a paper talking about how that was speculating the role of the superior colliculus in autism etiology. Mm-hmm. And I found it fascinating. I won't bore you with the ins and outs of the details now, but it made a lot of strong points, and I was like, oh, this is very interesting. I think it, it could be involved. Problems with visual attention and gaze fixation and its role in development and this and that. And it really wasn't very looked into, compared to cortical mechanisms and things like that, people looking at the cortex. And I just thought it was really interesting, and I was building up setup number 5, I think, for my new project, for a part of the old project. And I said to my boss, I was like, have you seen this paper? Maybe this could be interesting. What if I just try it? While I'm building the new setup, I can just try to get some mice from the group working on autism. Yeah. We can see. And then...
Wow.
And that's how it happened.
I think all of us who are doing a PhD, we are also struggling a bit with work-life balance. We are working really hard. Yeah. And especially when we reach the time when the failures begin to appear, and we are kind of, our mood goes down, we are feeling like the work is the only like thing in our life, it kind of brings us down. And I think it's something that sometimes we realize a bit too late, that we should really care about things outside of work to keep us going as well.
Oh, for sure.
So I wanted to ask you, like, what was it that kept you going, and what did you start to do to keep this balance, kind of like to have something else?
Yeah. Well, I don't know if you remember, but I think at almost the peak of my desperation with my lab work, of when nothing was working, I decided that was the perfect time to start the art club at work.
Yes. So I didn't realize this was the moment.
Oh, yeah. Because I think I was like, I need to do something else.
And you've always been very into art, right? You've always been doodling and drawing and...
I mean, yeah, always been a doodler. Like, for as long as I can remember, I've been drawing stuff. I think I was saying to my friend the other day, I remember when I think I was 7 and a boy in my class bought this like A4 page of Pokemon that I drew for like £2 or something. And I was like, oh, that was my first commission, you know, doing these stupid little cartoon doodles. But no, art is something that I've always loved doing. And I find it very therapeutic. I don't know, I like doing lots of different things. Like I like doodling and doing small like cartoons with pens, or I like doing very like swooshy, colourful paintings.
More abstract.
Yeah. I love portraits, I love like landscapes, I love still lifes, anything, and sort of all mediums. I think that's like my splurging buy, is I always buy myself a new paintbrush or a new crayon set or something. Which was really fun, but that definitely helped. And I decided to... Actually, I didn't have the idea for the art club, but I ended up running it through another series of weird events. But I was very thankful because it was really, really fun. And it was really nice to meet lots of people from work that I hadn't met before. And I think, you know, it was really nice coming every other week and seeing people and just chatting about random things and drawing or painting or doing something together.
Just not thinking about work so much. Being in a different space, right?
No, definitely. I mean, I always enjoyed sport and things. Like, I mean, I'd always go for walks. I love running as well. So that was definitely very useful during stressful times. I just go out and like run anywhere around Vienna. And then just friends. I mean, now it's a bit difficult during pandemic times.
Yes.
But before, you know, you just go for a beer.
What I find really cool is that you managed to somehow connect your outside of work passion, which is the art, with science and with what you do.
Aww.
And I would like to hear a bit more about this. I mean, I saw that you had a couple of different projects. Let's call it projects.
Yeah. Yeah, sure. I mean, I don't know, because to me, my doodling, I just think of it as like another tool that I have in a way. It's not even a tool, I don't know, but I think that's always how I've taken notes and things.
So does it help you think?
Yes, it definitely helps me think. But so it started like that, and yeah, I never really thought people would be that interested, but they really liked it, and then it sort of continued, and I was just doing fun things for my lab. Like my boss was like, "Oh, we've got a grant for this. Would you mind drawing a little logo?" And I was like, "Oh yeah, sure, why not?" Like, and then a friend from work got in contact about a really cool initiative that she was starting called the Wissenschafts initiative, and it's cool. It's a sort of form of science communication, and they're doing like YouTube videos and also on Instagram about different topics. And this first one is all about vaccinations, and yes, she reached out and was like, "Oh, would you like to help draw the animations?" And I was like, "Oh, I've never done an animation before."
I think it's a really cool way to, you know, explain to people, especially now in the times when the vaccinations are really a topic that is maybe misrepresented in media a bit. It's good to have something that is scientifically based to kind of help people to understand how it works.
I think it's definitely important, and I think, I don't know, but people don't take it as seriously. And I'm like, this is probably the most important thing, is like, what is the point of advancing knowledge if you're only advancing the knowledge for a very small
group of people? Yeah.
And honestly, I really love the aspect of taking these topics and really getting to the core nugget of information that you need, because I'm always like, how would I explain this to someone that has no idea about the sort of complex processes and things?
I think especially coming from the family where, like, my family is also like this. No one is really a scientist, and we often get into this discussion, at the table, when they're like, oh, we've heard about this, like, can you explain it?
Yeah.
It's not easy to tackle this, and I think this is something that is very overlooked. We should be able to communicate our work and our interest for science to people that maybe don't have as much knowledge as us, and to be able to put it in a simple way. I think the visual part of it is actually very helpful, like the drawings and the way to put it so that it's maybe a metaphor that is much more understandable, or making it animated so that people can really maybe it reaches them much easier.
I like to think so. It's just sort of more engaging as well. I think time is something that is it's very precious to people nowadays. I love reading articles like in newspapers and stuff, but like I was speaking to my boyfriend, he was like, the thing is, I don't think a lot of people have time to read long articles or these like press releases about new scientific breakthroughs and stuff. But everyone's like on their phone checking their Instagram or doing stuff like that.
It's easier to digest as well.
Yeah, and
probably.
Honestly, I think as long as you get the message across, that's
the important thing.
That's the main thing.
Did you like this kind of work, because this was a bit new?
Yeah, it was really cool. So what you have to know is, other than becoming David Attenborough, my second childhood dream was to be a Disney animator.
Oh yeah, so now you're getting there.
Basically, my dreams are coming true.
This is amazing.
Yeah.
I think, I don't know if you want to talk about this room for improvement part. Like, do you have anything in mind? Or we anyway, like, had pieces every now and then when we were talking about things that don't work so well.
There are definitely many things that could change in the system. Let's just put it that way. But I think one thing that I saw very recently, like just the other day, was an image online of how supplementary figures have changed over time. So it was just looking at like a standard supplementary figure in a paper from 2007, and then then like 5-year gaps until now, and it's changed from like one graph and 2 images to like a 25 image like subplot thing. Yeah, exactly, which is just insane. And then someone commented about the fact that what if we did a rolling upload of research instead? And I'm sure there are many downsides to this, but it was just the first time that I'd sort of thought of that idea. I'm sure other people have brought it up at other times. But it was just really interesting thinking of a completely different system where, you state beforehand, like, I will be working, like, I'm planning on working on this, and then you sort of upload it. Like how we have progress reviews every 3 months, you just upload where you're at with it. Again, I have no idea how, like, what happens with people doing the same thing. And I don't know, I think it's just strange how we give credit to people nowadays, like this idea of scooping, and
it affects the progress of science kind of negatively, because people just feel like there's a lot of competition where actually collaboration could bring us the answers faster.
Definitely.
But I think the whole system is so deeply based on the publishing, and it's something that is very idealistic, but I think it's a beautiful idea. It's
an
just
interesting thought, child.
No, I'm not trying to be condescending. What I mean is, like, I think we as like students, we still think that this is really cool, because we see that this just would help advance everything faster. We would know what somebody else is doing, and you could.
That's the thing. You could do the same thing, and people should do the same things.
And then you could check if this works.
Exactly, because like I've done, like dabbling in many different techniques that I've been doing. I've done things which people say should definitely work, and I'm like, it hasn't worked. And then I talk to other people, and they're like, oh no, it also didn't work for me. And I'm like, why are people floundering around spending so much time and wasting so many animals and materials and time on these things? And if everything was just more open, but this is, I think, very.
I mean, I think it touches also another point that is very important, but also something that I think is an ongoing discussion, which is publishing of the negative results. Mm-hmm. So, how informative it could have been if people would just also talk about the things that didn't work.
Yeah.
Or if there was a repository, I think, where, you could just check and maybe then you do it. But if you see that it's not working for you as well, you're kind of feeling a bit better about this, more certain. And I think there have been voices about this. I'm not sure if this is really doable, but...
That's the thing. I don't know. It almost needs very specific templates that you fill in, of exactly what you have done. I just think because the variability within, obviously things change so much. There are so many variables that
the environmental variables
that you need to put in order to compare across groups and things.
I think it may take a while, but science is also changing a lot. As you said, the supplementaries were smaller, but I think the reason why they are getting bigger is because people are really paying much more attention to to kind of have enough evidence, and they are not maybe so trusting anymore, which can be considered a bad thing. But on the other hand, I think it makes it easier for the research to be reproducible and to be properly validated, which is, I think, something that is also important. Okay, so last thing, which I think is a fun thing.
What?
And because you didn't listen to the other episodes, you actually don't know. So I want this to be kind of a recurring thing and a question that I ask to everybody, because I find it very interesting to hear what people think. If you had unlimited money, what is the experiment that you would like to do?
Oof.
But another question that I think I know the answer, partially at least, is if you could have a dinner or a tea with someone who inspired you, someone who is a cool scientist for you, or someone that you just would like to meet. So I think in your case the answer is very easy, but maybe you have another, except for Attenborough, who I think would be your first and primary choice.
I mean, there are so many people. That's
but did I guess well?
Yeah, of course. I mean...
So would you like to have a dinner with him, or would you like to go on an adventure with him? What would be your biggest dream?
No, I mean having maybe a tea in the middle of the rainforest or something. In a thermos flask. That would be cool. I think David's sort of getting a bit old now, though, so I think I would feel bad making him walk through the rainforest. Many things, and God, the experiment. That's a tough one. Infinite,
you're not limited by money. You can have any supplies.
I mean, something that I think would be really cool is just using different model organisms, which would require lots of money to try and, understand them. But I think a mouse is very cool, and it obviously displays this behavior of avoiding predators. But there are lots of animals in the animal kingdom and insects and stuff that have a lot more specialized behaviors that I think if we're trying to understand certain things about the brain, you can you have to look at where the animal is and its niche and what it's specialized for.
So do you think that the predators would also have some sort of responses that actually are processed by superior colliculus?
So, for instance, in birds, the predators of the mice, their optic tectum is what the superior colliculus is called in birds. It's huge, massively more important than in the mouse. They have a huge optic tectum, and it's their main visual center. In the mammals, for instance, it's 7-layered, the superior colliculus, and I think it's 14 or something in the bird. I could be wrong, but it's a lot.
But is it known that it's connected to their flight?
Yeah, exactly. So they have a very strong they have great vision, birds of prey. They have to spot their moving prey from miles away, a long distance away. So they are definitely a lot more specialized. So I think moving to
So you would like to
super cool.
try record from birds? And
I mean, people do. There are lots of people working on birds, but the problem is that you don't have the same genetic tools in birds. So maybe, you know,
Yeah, this would
improving
be
the
really
genetic
cool.
tools in bird species. I was talking the other day about looking at spatial navigation in amphibians, like things that work in both water and on land, and how the spatial maps
do
are
you think
different.
that because of the water it's much more 3D? Or
Yeah, that's the thing, you know what I mean? They go from like a 2D environment to a 3D environment. Like, how do they map that? But it's you can't really like record in water. That's
that's
the problem.
true, that's
the people
true.
only do it in zebrafish because they're transparent when they're larvae.
Yeah, you can see.
Yeah. See, so many problems that need to be addressed.
So, okay, recording from birds and then having a tea with David Attenborough.
In a thermos flask in the rainforest.
Yeah, definitely. This sounds really cool. We never know, right? I mean, rainforest part, I would be a bit skeptical. Like,
Okay, fine. I'll choose some nice like wooded forest in England.
that's much more doable, I think.
Yeah, I think so
Still,
too.
considering like all the factors. So
Yeah.
I wish you manage, or even just like, have you ever heard him speaking live? Actually,
No.
no. Like I know that his engagements are now like very limited, but just yeah, this would be something cool. Thank you, Laura.
No,
this
thank
was
you.
Very inspiring.
Oh, it was super
Conversation.
fun.
And I'm looking forward to seeing more of your art. Actually, be sure to check out Laura's art, and I will link it
Stop it.
in
You're
the
too
post.
kind.
Okay.
Okay. Thanks a lot.
Thanks.